Laboratory Manual in General Zoology. Exercise number 1. Animal cells and tissues. Introduction. The cells theory states that all living organisms are made up of cells. Cells are the basic structural and functional units of life. Cells differ in size, shape, and function. These variations are correlated with the functions they perform. According to structure, cells are generally classified either as prokaryotic or eukaryotic. According to function, animal cells are either somatic or reproductive. Multicellular organisms are made up of several cells. Cells that perform the same functions are called tissue. Cells of a tissue may be of several kinds. Tissues are in turn grouped together to form organs. Organs group together to form a size, and all these comprise an organism. Objectives. Number 1. To identify the commonly visible parts of an animal cell. Number 2. To describe the major parts of cells and their functions. And number 3. To identify and describe characteristics of animal tissues. Materials and methods. Light compound microscope prepared slides of the following cardiac muscle tissues. Simple squamous epithelium, smooth muscle tissues. CS of kidney tubules, simple cuboidal CS of a bone. Epithelium, CS of a hyaline cartilage, CS of frog's intestines or stomach, human blood smear. Simple columnar epithelium, also used for smooth muscle epidemic. Adipose tissue, XS frog's ovary, human sperm smear. Number 1. Somatic tissues. Epithelial tissues. Take note of the compact arrangement of cells and presence of basement membrane. Notice this thin and permeable membrane that binds the epithelium together and separates it from the underlying connective tissue. Observe carefully if the focus epithelium is composed of one layer of cells or simple, or two or more layers or stratified. Under the close inspection, you will notice that epithelial tissues have no blood supply. Since they are avascular, nutrients and oxygen are supplied through diffusion from underlying tissues. Observe the following simple epithelia. A. Simple squamous epithelium. Take note of the thin, fat, and irregularly shaped cells. They line the heart, blood, and lymphatic vessels, body cavities, and alveoli, air sacs of the lungs. Examine a prepared slide of simple squamous epithelium. Whole mount under the LPO and HPO and identify all visible parts. B. Simple cuboidal epithelium. Examine a prepared slide of kidney tubules. Use the HPO to closely examine the wall of the tubules. Take note of the cube-shaped cells that line the kidney tubules. This type of cells can also be seen lining the ovaries and secretory parts of certain glands. C. Simple cuboidal epithelium. Examine a prepared slide of kidney tubules. Use under the LPO. Locate the finger-like folds called villi of the small intestine. These folds are lined with columnar cells. Look for elongated cells with the nucleus generally near the bottom and often ciliated on the outer surface. Turn to HPO and carefully examine individual columnar cells of the villi and identify all visible parts. Number 2, connective tissue. Carefully observe the features of the tissue that makes it bind parts together, fill in space, and provide support for other types of tissue. Notice that these tissues have fewer cells but with more intercellular substance, matrix or ground substance, than the epithelial tissues. The matrix is variable in type and amount. It is one of the differentiating features of the various types of connective tissues. In vertebrates, connective tissue proper includes both loose connective tissue composed of fibers and fixed and wandering cells suspended in a syrupy ground substance, and dense connective tissue, tendons and ligaments, largely composed of tightly packed fibers. Other types of connective tissue include the vascular tissue, blood, lymph, and tissue fluid. Vascular tissue is found in a fluid ground substance and called the plasma. A. Cartilage. Cartilage is a semi-rigid connective tissue embedded in a gel-like matrix. Examine a cross-section of a hyaline cartilage under LPO and HPO. Look for these parts: chondrocytes or chondriocytes cartilage cells. Lacunae, singular, lacuna, cavities containing the cartilage cells. Bone tissue. Examine a cross-section of the bone. Under the LPO, carefully observe the different structures and their arrangement. Take note of the calcified matrix with calcium salts organized around collagen fibers, arranged in concentric rings lamellae under HPO. Focus on a single Haversian system and identify all the parts. Osteocytes bone cell. Lacunae, cavities where bone cells are lodged. Canal cord, small canals that interconnect the lacunae to distribute the cellular materials. Haversian canal, the central canal with blood supply, vascular. Hosted or Haversian system, entire elongated cylinder unit of the rings. The osteon is the circular and foundational unit of a bone. B. Blood. Take note that in fresh specimen, this tissue is made up of the fluid portion, plasma, and the formed elements, cells, corpuscles, and fragments. These cells are number one, red blood corpuscles. Number two, leukocytes or white blood cells. And number three is thrombocytes or blood platelets. Examine a prepared slide of a human blood smear and a frog blood smear. Focus then under the LPO and HPO. Take note of the differences in terms of shape and size of cells and the presence of or absence of a nucleus in each cell. C. Adipose. Examine a prepared slide of adipose tissue under the LPO and HPO. Look for large soap bubbles-like structures or signet rings with nucleus and cytoplasm posed to one side. This is a loose connective tissue that stores lipid, cushions, supports, and insulates organs and acts as filler tissue. Number 3, muscle tissues. Observe that muscle tissue is composed of elongated cells called muscle fibers, specialized for construction. This specialized function of construction is exclusive for muscle tissues. Muscles are the most common tissue in the body of higher animals. A. Skeletal muscle tissue. Secure a prepared slide of a treated skeletal muscle and examine it under the LPO and HPO. Muscle fiber or muscle cell, elongated, unbranched, and multinucleated fibers. Steering, transverse alternating dark and light bands. B. Cardiac muscle tissue. Examine the cross-section of the cardiac muscle under the LPO and HPO. Note the light and dark bands like those exhibited in skeletal muscles. Observe that the fibers seem to unenclose with the adjacent ones, interconnecting branched fibers. In the intervals along the fibers, find thin, dark intercalated which is characteristic of the cardiac muscles. C. Smooth or visceral muscle tissue. Examine a cross-section of a frog's stomach under the LPO. Move the slide to the circular layer of muscle. Using the HPO, focus on a spindle-shaped fiber which has a nucleus at the center of the widest portion of the cell body. Number 4. Nervous tissue. The nervous tissue is made up of two elements, the neuron or nerve cell and neuroglial cells. Neurons are concerned with the transmission of impulses, specialized to receive and transmit impulses from one part of the body to the other parts. Refer to the figure of a neuron. Take note of the following parts. Number 1, parts of the nervous tissue is cell body. Contains the nucleus and the thread-like process. Number 2 is the neurema. The neurema is a cell membrane. Number 3 is dendrites. Dendrites is the process that conduct impulse towards the cell body. And number 4 is axon. Axon is conduct impulse toward from the cell body. Number 2, reproductive tissues. The reproductive cells are produced by the gonads. The male gonads or testes produce the spermatozoa or sperm cells. The female gonads or ovaries produce the ovum or egg cell. A. Ovum or egg cell. Examine under the LPO the frog's ovary. Focus one folide with cells at different stages of development and take note of the following parts. 1. Oocyte. True ovum surrounded by follicle cells. 2. Granulosa cells. Multi-layered cohort or folide cells surrounding the ovum that secrete the fluid that accumulates in the extracellular spaces and but that oocyte. Number 3 is zona peloda, a cellular membrane between the oocyte and granulosa cells. Antrum, space between the ovum and granulosa cells. 5 is Graafian follicle, a folide with large antrum. Make a sketch of the scene ovary on the space provided for in a worksheet. B. Spermatozoa or sperm cells. Examine a prepared slide of human sperm cells under HPO. Identify the following. Acrosome, cup-like and found at the anterior portion of the head of a sperm. Number 2, head contains the nucleus with the genetic materials. Number 3 is contriol present in the basal plate of sperm. Head give rise to the axoneme of the flagellum. Number 4, mitochondria, site of ATP production to support the cell motility. Number 5, middle piece or mid-piece, portion that contains the mitochondria. And number 6 is flagellum or thorn, the long whip-like structure used for movement. Exercise number 2. Kingdom Protista, Subkingdom Protozoa. Introduction. The kingdom Protista or Protoctista, subkingdom Protozoa are unicellular, microscopic, animal-like organisms. These life forms provide materials to understand the basic life activity of multicellular animals. They occur in all types of habitats. Many are parasites, others are free-living, some are solitary, and others are colonial. Below show the common group of protozoans. Pilium sarcomastigopora. Locomotory structures are either flagella or pseudopodia, usually with only one type of nucleus, no spore formation. Subphylum Mastigopora, class Zomastigopora, move by means of flagella or singular flagellum, predominantly parasitic. Example is Trichomonas, Trypanosoma, and Lesmaria. Subphylum Sarcodena. Movements Move by means of pseudopodia or singular pseudopodium, present free-living or parasitic. Class Lobutia, pseudopodia for movements and food gathering, usually uninucleate. Most are free-living, some parasitic. Example is Amoeba intimiaba histolytica. Phylum Apicompliza, cilia and flagella absent, except on certain reproductive stage. Cysts often present parasitic with apical complex used to penetrate host cells. Plus sporozoa. Spores of oocytes that are typically present contain infective sporozoites. Flagella present only in microgametes of some groups. Pseudopodia ordinarily absent. If present, they are used for feeding, not for locomotion. Have life cycles involving one or two hosts. Example is Plasmodium and Toxoplasma. Phylum Ciliopora. Cilia or ciliary organelles present in at least one stage of life cycle. Has two types of nucleus. Most species are free-living. Many are commensals and some parasitic. Example is Paramecium class Celeta. Objectives number 1, to study the cellular organelles of protozoan. Number 2, the characteristic features of protozoans, and number 3, to classify representative protozoans with different modes of living. Materials and methods. Light compound microscope, prepared slides of amoeba, paramecium, plasmodium. Slides and overslips high in fusion culture cotton fibers. Procedure A. Amoeba, also spelled amoeba plural, amoeba or amoebe. These are generally parasitic, though some forms are free-living. Number 1. Get a, get a prepared slide of amelia and examine under the LPO and HPO. Identify the visible organelles. Number 2, get a draft of the hay infusion culture and prepare a wet mount. Examine under the microscope, guided by the morphology of amelia as seen in the prepared slide. Look for an amelia mimba. Number 3, note the general appearance, colors, cytoplasmic organelles. Letter B, paramedium, a free-living protozoan. Number 1, prepare another wet mound from the hay infusion culture. Minimize the movement with the use of cotton fibers. Focus on the LPO. Observe how the animal moves. Avoidance reaction. Number 2, shift to HPO. Determine the oral grooves and other visible structures. Observe the movement of the cilia on the cell surface. Number 3 is observe other structures inside the cell and all visible parts. Before also view a prepared slide of paramecia under LPO and HPO. Label the drawing on the next page. Use the word bank below. Exercise number 3. Introduction. Poriferans of sponges are the lowest form of multicellular animals. The body has many pores called ostia, singular or ostum, and consists of aggregates of cells supported by a framework of microscopic roots called spicules and collagen fibers called spongen in some species. Adult forms are sessile attached to the substrate, but their larvae are free swimming. Sponges are aquatic organisms, majority of which are marine but with some freshwater species. These animals reproduce asexually through budding, fragmentation, and gemellous internal buds. However, they are also capable of sexual reproduction involving sperms and oocytes that are usually produced at different times. Kingdom Animalia or subkingdom Parazoa. Phylum Porifera, sponges. Classes of phylum Porifera based on their skeletal support, primarily spicules. Class Calcarea have calcareous spicules, example Grantia or Scypha. Class Hexactinellida have 6 ray siliceous spicules, example Venus flower basket. Class Demospongiae siliceous spicules spongean or both, example Spongia, bot sponge. Class Sclerospogli coralline sponge. 3. Basic level of sponge construction showing the canal system. Number 1 is Ascon, simplest and less common sponge body from form. 2. Cyclone, sponge wall appears folded radial canals lined with chorocytes. Number 3. Leocon, most complex canal system, has many chambers and extensively branched canals. Kingdom Animalia, subkingdom Eumetazoa. Phylum Cnidaria, hydroids, jellies, corals, and sea anemones. Eumetozoans are multicellular animals that have throat tissues organized for specific function. The simplest form of metazoans include the cnidarians. They exhibit radial symmetry, wherein body parts are arranged around the oral aboral axis. They are diaplastic organisms with two germ layers, the ectoderm and epiderm, but a mesoderm may also be present in some. Mesoglea, a gelatinous matrix, is found in between the layers may al- between the layers. The sac-like body is closed at the end and open at the other end. Their unique characteristic is the presence of stinging cells called cleidocytes, which stinging organelles, the nematocytes. These organisms live solitary or in colonies. The process is exhibited in having individual types that are attached, called polyp, while the free-swimming body that form is called medusa. Colonial types have a alternation of sexual and asexual generation. They are classified into class Hydrozoa, can be found in but both marine and freshwater habitats have polyp and medusa form. Example: Hydra polyp form, Obelia polyp, and Medusa polysalia. Pisalis, Portuguese man or war, polyp, and medusa. Class Scupozoa through jellyfish, medusa stage dominant all marine swimming. Example: Aurelia. Class Anthozoa, polyp stage dominant all marine, may be colonial or solitary. Example: coral Acropora fungi, Toby paramusica, sea anemone, Metridium. Class Cubozoa, solitary polyp stage reduced all marine, also called box jellyfish. Example: Chronix fleckeri, sea wasp. Objectives: number one, to study and compare the characteristic features of poriferans and cnidarians. Number two, to describe the structural feature of representative animals under this phyla. Materials and methods: light compound microscope, prepared slides of Grantia sponge follicles or spongine hydra plant budding Obelia. Binocular design microscope, preserve specimen of different sponges, Eulyptilla sp. Venus flower basket, corals, sea anemones, or jellyfish. Phylum Porifera. Number one, external anatomy of sponges. Obtain and observe a dried sample of Venus flower basket. Touch the glass skeleton and silicon that characterize that class Hexactinida. Examine the base of available sponge. Are they attached to some object, or do these show evidence of having been attached? Secure a prepared slide of sponge spicules. View under LPO and then take note of the different forms. Draw what you have seen. Number two, canal system of the sponge. Examine prepared slide of Grantia under LPO. Move the glass slide to identify the following. Number one is ostia pores that serve is as incurrent openings. Two, in current canal, water route to the spongicoil. Number three is spongicoil gistral or internal cavity. Identify the type of canal system of your specimen. Indicate in the drawing the water route of this type of canal system. Phylum Congenedria 3 Hydra. Examine and prepared slide of Hydra and solitary Hydra under LPO and compound microscope or under the dissecting microscope. Identify the following parts of the labeled in your drawing. Basal disc for attachment to the substrate, hydrant, oral end, and polyp bearing of the mouth and tentacles. Hypostome, colonial elevation containing the mouth part. Bud, asexually produced outgrowth. Thank you.